We have been learning about movement in protozoan, specifically movements using appendage. And in a previous video, we had seen how they move using a flagella. In this video, we are going to be focusing on how protozoans use cilia for various purposes. Before we get into the video, I'll show you what we are talking about. This is a paramecium that is moving about in a water medium. And you can see that there are these very tiny hair-like structures that are found all around it, right? And they are moving in a specific pattern, and that is making the organism also move in a certain fashion. The organisms that possess a cilia are called as the ciliates. Primitive ciliates like Paramecium, they have cilia on the entire body, whereas advanced ciliates like Vorticella, it is restricted to the peristomial region. So this is the mouth region through which the food enters into the organism. There are certain groups called as the suctorian protozoan ciliates. So the adults of these organisms can suck the food out of its prey. An example of this is Acineta. So in this, you see these cilia-like structures only during the juvenile phase. In the adult, these cilia develop into tentacles with suctions at the end that helps them in feeding. Eukaryotic cells also can possess cilia. They might look something like this. These cells are columnar cells, and the cilia is present on their apical surface or the upper surface. So these are called as ciliated columnar epithelium. And there are few places where these could be found. They are found in the respiratory tract, meaning your nasal cavity, your trachea, bronchi, etc. Genital ducts, which is your fallopian tube, the tube through which the ovum moves from the ovary into your uterus, and finally, ventricles of the brain. These are spaces within the brain and spinal cord where there is circulation of cerebrospinal fluid. The cells lining these spaces are ciliated columnar epithelium. The cilia move in a certain fashion, so the movement can allow for circulation of the CSF. Cilia has the same origin as flagella. If you recall in the video on flagella, we learned that flagella originate from organelles called as the basal granule. But their uses could be slightly different from flagella. Organisms use cilia for locomotion, food collection, and some of them act as sensory structures that detect changes in the environment. Let's see an example of how a paramecium uses its cilia to obtain food. So this part of the paramecium is called as the gullet or the mouth opening, which leads into the structure called as cytopharynx. So this is a small tube-like structure through which the food enters into the body. This is the contractile vacuole that helps in processing this entire digestion. Let's say there are some food particles that the paramecium has seen hanging around its gullet. Look at the movement of the cilia near this mouth area. Okay, so what happens is that the cilia, they start beating in a certain fashion, and this creates waves of water moving into the organism. And as the water moves in, you can see that the food particles are also moving into the organism. So what is the paramecium doing here? It is using the cilia to change the movement of water around it so that the food can enter into its body. Looking at this, the next question should be, how do all these cilia move in sync? See, we have seen a 2D picture now, but the entire surface of the paramecium is covered in cilia. So how are all these coordinated? So let's take a look. Remember that these protozoans are surrounded by a structure called as the pellicle. Yes, pellicle replaces their cell wall. But this pellicle is not just a single layer. The pellicle actually has 3 layers within it. The outermost layer of it is what we call as the cell membrane. Okay, so this is what we see from the outside. Now, the middle membrane is called as the outer alveolar membrane, and the inner membrane is called as the inner alveolar membrane. So here you can see that they are extensively folded and there are few spaces that are present between them. The kinetosomes or basal bodies from which the cilia or flagella originate are present in these grooves. So all these structures together are the pellicle, and below the inner alveolar membrane you find the cytoplasm. And from these grooves which contain basal body, you have the cilia coming out. So this is how the structure appears from the outer surface. But in order to understand the synchronized movement of cilia, you have to look a little inside them. So let's imagine we have a ciliate. This is just a representative diagram for an organism. Okay, and let's assume this is the anterior or the front portion and posterior and the back portion of the organisms. It is seen that the placement of the basal body or the basal granule is not random, but are present in transverse and longitudinal rows in this particular manner. And now let's again focus into a small section of it. This is a smaller section, and again we have anterior and the posterior positions. The circular structures you see here are the basal granule or kinetosome, and they are connected to each other by three fibrillar structures. So you don't have to remember all the structure. If you remember one, that is good enough. And these connections are again not random. They are connected in a transverse as well as in a longitudinal manner. The first of these connections is called as a transverse microtubule. Now this is like a ribbon of microtubule that extends to the right side of the basal granule. The second one is going to connect them longitudinally. It is called as the post-ciliary microtubule. You don't have to worry about the name here. You just have to know that this particular ribbon of microtubule is going to extend posteriorly. So it is going to connect this one with the basal granule behind it. And then finally, we have the most important one. These are striated microtubules that extend anteriorly, means towards the front. And that too, they run from the base of one of the basal granule to the right side of the basal granule at the front of it. These are called as the kinetodesmal fibrils. Now, this is important. These kinetodesmal fibrils occur as a cord or rope-like structure that is bundled towards the right side of a basal body, and they become kinetodesmata. This is also important. Why? Because these form the units that actually help in controlling the movement of cilia. So when you bring together longitudinal kinetosomes, so these rows of basal granules, they combine with the kinetodesmata, they form a unit which is called as a kinety. Even if this is too much to take in, you just have to remember that the longitudinal arrangement of basal granule along with kinetodesmata gives the unit called as kinety, and that is going to help the cilia move. Kinetes form what we call as the infraciliary system. This is the arrangement which is responsible for the cilia moving in a certain way. So this is like the basic structural framework, and if the cilia are not attached this way, it would be very difficult for them to move in a certain uniform fashion. Now this infraciliary system, it's just the cilia. It's not actually controlling it. It's just the structural system, right? Infraciliary system combines with an organelle found near the cytopharynx called as the motorium. For an organism like Paramecium, intaking food is the most important for its survival, right? So the way the body moves and the cilia moves is directly associated with finding food, and hence motorium is located near the cytopharynx. Motorium receives and processes the signals that it gets from the cytopharynx and then uses the infraciliary system to move the cilia in a certain way. So together they form what is called as the neuromotor system in Paramecium, which controls and coordinates cilia. The final way in which protozoans can move is using the contractile protein, which is called as a myoneme. These are also found below the pellicle. They are found in different types of protozoans. You can see them in flagellates, you can see them in ciliates, and you can even see them in epicomplexans. So this is the group where Plasmodium, the malarial parasite, belongs to. Let's take a flagellate. So in Euglena, this particular myoneme is found just below the pellicle in this particular fashion. The movement of myoneme is very different from cilia. In cilia, it's more of a smooth movement, but in myoneme, it's more of a sudden, forceful move.